I'm Christopher Rowley, and I'll be presenting a video summary of our recent paper in the Journal of the American Chemical Society titled, Why Can Hydrogen Sulfide Permeate Cell Membranes? Our objective was to examine a result that had been reported by John Matai and PNS in 2009, which showed that hydrogen sulfide does not need a protein transporter or facilitator for it to cross cell membranes, and can instead cross really quite rapidly just through a process of passive diffusion. Quantitatively, they determined that the permeability coefficient of hydrogen sulfide must be at least 0.5 centimeters per second. And in contrast, something like water has a relatively small permeability coefficient of only around 2.6 times 10 to the minus 5 centimeters per second. And this is a little bit surprising because hydrogen sulfide is a structural analog of water and also has a significant permanent dipole moment of almost 1 to pi. To examine this process, we use a solubility-diffusion model that was developed by Marink and Berenson. What this model does is to calculate the permeability coefficient by integrating an expression that depends on the Gibbs energy profile and the diffusion coefficient profile across a transmembrane axis, in this case indicated as Z. And we integrate between values on this axis between Z1 on the interior of the membrane and Z2 on the other side. And using computational chemistry, we can calculate these Gibbs energy and diffusion coefficient profiles of these solutes at every point along this axis, and this provides us with a strategy to calculate these permeability coefficients. In our simulations, we used a fully polarizable molecular mechanical model of our bilayer and water and solutes surrounding it. So the DPPC lipids, water, and solutes were all described using a polarizable model that was capable of including the effect of induced polarization. We also used a relatively large simulation cell that was 64 by 64 by 96 angstroms cubed. The water layers above and below the bilayer were comprised of a total of almost 10,000 water molecules. The bilayer itself was comprised of 132 DPPC lipids, and this gives us a total of around 75,000 particles. To calculate our Gibbs energy and diffusion coefficient profiles, we perform molecular dynamics simulations where the solutes, and in our case water and hydrogen sulfide, were restrained at different points in solution and crossing the membrane along this transmembrane axis Z. And so we performed a total of 40 simulations for both hydrogen sulfide and water at various points along this profile. In order to capture the effect of induced polarization, we used the Drude polarizable force field in our simulations. This is a model that mimics the effect of induced polarization by adding extra particles, known as Drude particles, that are charged and harmonically tethered to their parent heavy atoms. And because these particles are charged, they move in response to the electric field of the environment. And in this example, we have a positively charged potassium ion moving around a water molecule, and we see the dipole moment of the molecule change due to the movement of this Drude particle in response to this electric field. And so this provides a relatively efficient way of including the effects of induced polarization in our simulations, and also has some practical advantages. One practical advantage is that models that are based around the TIP3P water model actually are really quite bad for calculating diffusion coefficient profiles, and will typically overestimate the rate of diffusion by up to a factor of 3. In comparison, the Drude polarizable model was parameterized to perform well for rates of diffusion, and so tends to be quantitatively accurate for diffusion coefficients. Further, because the membrane permeation process corresponds to a process where a solute is moving from a very polar environment of water and through this nonpolar membrane, these kind of simulations allow us to monitor the change in polarization of the solute as it crosses the membrane. The limitation is that we can only perform these simulations on systems where there's parameters available for it. So we use the SWIM4-NDP water model that was developed by Guillaume Lamoureux and published in 2006. We used the DPPC lipid model that was published in 2013. And we developed our own model for hydrogen sulfide that was republished in the Journal of Physical Chemistry B in 2013. So I'll first show the results of our transmembrane diffusion coefficient calculations. So this plot shows the rate of diffusion of water and hydrogen sulfide as a function of the z-axis crossing this membrane. What we see is that when the solutes are in solution, they have relatively high rates of diffusion. But when we go through the head group area, we have a really quite large drop-off, and we see relatively slow diffusion. When we enter the interior of the membrane, where the lipid tails are, we see, again, relatively large rates of diffusion. And we can explain this based on the idea that in the membrane interior, the tails of the lipids behave more like a fluid, and so they have a lot of configurational freedom, and it's essentially a fluid-like environment again. One interesting to note here is that these diffusion coefficient profiles of hydrogen sulfide and water really aren't that different, so they're only no more than a factor of 2 different at any point. And so this is far smaller than the at least 20,000-fold difference that was observed experimentally. And what's more is the rate of diffusion for water is actually somewhat faster for the most part than that of hydrogen sulfide. And this is actually to be expected because hydrogen sulfide is a larger molecule and therefore experiences a greater degree of friction. Where we see a much larger difference is in the Gibbs energy profiles of the two solutes. And here we see hydrogen sulfide has a really negligible barrier and in fact has a negative Gibbs energy profile through the membrane interior relative to the solvent, indicating that it can even partition inside the membrane to some degree. Water, on the other hand, has a really quite large energetic barrier for Gibbs energy barrier for crossing the membrane. And we can rationalize this by noting that in solution, water will have very strong hydrogen bonding interactions with other water molecules. But when it passes through the membrane interior, those interactions will be broken, and the water will only have relatively weak London dispersion interactions with the membrane interior. Using these data, we calculated the permeability coefficients of water and hydrogen sulfide using the solubility-diffusion model. For water, we actually saw a really extraordinarily good agreement with experiment where we actually matched the experimental value of 2.6 times 10 to the minus 5 centimeters per second exactly. This is somewhat fortuitous because there's certainly enough approximations in both the solubility diffusion model and in our model, our polarizable molecular mechanical model, we would expect a greater degree of error, but nevertheless gives us some confidence that our calculations are doing a reasonably good job of describing this membrane permeation process. In contrast, sure enough, we see a much higher rate of permeability for hydrogen sulfide. So we calculated a permeability of almost 12 centimeters per second, and this is even higher than the experimental value that had been determined experimentally, although the experimental value was just a lower bound. And so we are predicting that, in fact, the rate of permeation of hydrogen sulfide may be even faster than had been kind of determined experimentally. This result can be understood by going back to a very old theory in membrane permeation, which was Overton's rule, who noted that more hydrophilic solutes tended to permeate relatively slowly, whereas more hydrophobic solutes tended to have faster rates of permeation. This can be quantified by correlating the water hexadecane partition coefficient with the rate of permeation. And in a study by Walter in 1986, she showed that there was really an excellent agreement between these two parameters. And this essentially reflects that the solutes that tend to partition into the hexadecane phase and have large partition coefficients would be relatively good at permeating into the interior of the membrane, which is a lipid-like tail environment that is chemically similar to the environment of a bulk alkane like hexadecane. Whereas more hydrophilic solutes that tended to partition towards the water phase had relatively low rates of permeation because these solutes would have a smaller tendency to exit the aqueous phase and enter this hydrophilic interior of the membrane. And when we put these, both our calculated and the experimental data, into this data set, we see that we're actually not far off. And so our predicted rate of permeation is more or less what we would expect. And it simply corresponds to a relatively high partition coefficient, hexadeciane partition coefficient for hydrogen sulfide, and therefore a relatively large rate of permeation. This high permeability of hydrogen sulfide can be understood essentially based on its low solubility in water, and it has actually a really quite weak hydration free energy. We had investigated this in some detail in a previous study by our group published in the Journal of Physical Chemistry B. In a study, we examined the solvation structure of hydrogen sulfide in water, and what we noticed was that there was no very strong or permanent hydrogen bonding interactions between water and hydrogen sulfide. And more commonly, we would see the water surrounding hydrogen sulfide hydrogen bonding with each other and essentially encapsulating the hydrogen sulfide in a kind of clathrate-like hydrophobic shell. We can understand this by noting that sulfur is a second row element, so it has a large atomic volume, and it has a relatively modest polarity. So the polarity of the sulfur hydrogen bonds is small relative to that of water, and so it cannot form particularly strong hydrogen bonds with water, and so the water surrounding hydrogen sulfide would tend to hydrogen bond with each other instead. Essentially, what this gives us is just purely hydrophobic solvation of hydrogen sulfide, which allows it to exit aqueous solutions and pass through the membrane without incurring any kind of significant enthalpic penalty due to the loss of interactions with the aqueous solution. To conclude, I'd like to thank the Memorial University of Newfoundland, the Research and Development Corporation of Newfoundland and Labrador, the NSERC Discovery Grant Program, and AceNet and Compute Canada Consortia for providing our computational facilities.